Welcome to From DNA to Protein, where we uncover how cells turn genetic code into real life action. Think of DNA as the cookbook, RNA as the delivery service, and proteins as the finished meal. Today we will explore transcription and translation, the two steps that make this possible. By the end, you'll not only know what happens, you'll remember why it matters for your exams and maybe for your cells too. DNA is like the hardcover copy of your biology textbook. Precious, sturdy, and not meant to leave the library. RNA, on the other hand, is the photocopy you take to class. It's single-stranded, uses ribose sugar instead of deoxyribose, and swaps thymine for uracil. This simple step is surprisingly important. It lets enzymes distinguish between permanent storage and working copies. This is called the central dogma of molecular biology, the one-way flow of genetic information. DNA is transcribed into RNA, which is then translated into protein. It's like going from an idea, DNA, to a draft, RNA, to a final product, protein. Simple enough, until you realize how many molecular machines have to cooperate perfectly for this to happen. Transcription is where the cell makes a working copy of the gene it needs. RNA polymerase unzips the DNA at the right spot, the promoter region, and starts building a strand of mRNA, matching A to U and C to G. This mRNA will eventually leave the nucleus to meet the ribosome. Think of it as writing down a single recipe from your huge genetic cookbook. RNA polymerase is the unsung hero here. It's the enzyme that does the reading, writing, and proofreading, all while your DNA stays safely inside the nucleus. It binds to the promoter, reads one DNA strand, and builds the complementary RNA strand in the 5' to 3' direction. Without it, your genes would stay silent, literally. After transcription, the mRNA is like a rough draft, full of notes that don't belong in the final version. The cell's editors, called spliceosomes, remove introns, or non-coding regions, and stick together the exons, or coding sequences. It also adds a 5' cap and a poly A tail to protect the message. It's molecular publishing at its finest. Translation is where the language truly changes, from nucleotides to amino acids. The mRNA leaves the nucleus and binds to a ribosome. Then, tRNA molecules show up carrying amino acids, matching each mRNA codon with an anticodon. It's a fast-paced molecular assembly line, and nobody wants to drop a part. tRNA is like the delivery driver who shows up with exactly the right package. Each tRNA carries a specific amino acid and recognizes a codon on the mRNA using its anticodon. Once matched, it drops off its amino acid and heads back for another one. Reliable, precise, and totally underappreciated, just like your group project partner who actually reads the directions. The genetic code is a three-letter language. Every codon of three nucleotides translates to one amino acid. AUG always starts the party, and it's known as methionine, while UAA, UAG, and UGA tell everybody it's time to go home. Those are the stop codons. This code is nearly universal, from bacteria to bananas, which means the same molecular rules apply to you and your lunch. As amino acids link up, the growing polypeptide chain folds into a specific 3D shape, guided by hydrogen bonds and interactions. That shape determines everything: structure, function, even enzyme activity. One wrong amino acid can change everything, as in sickle cell anemia. So, yes, even at the molecular level, folding laundry is important. Students often mix up where each step happens. Remember, transcription is in the nucleus, translation in the cytoplasm. Codons are on the mRNA, anticodons are on tRNA, and the flow of information goes one way: DNA to RNA to protein. Also, in any diagram question, label the 5' and 3' ends correctly. It's free marks waiting for you. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.